So welcome to video 2 in of course G07 on phylogenomics. What we're talking about now is how to read a phylogenetic tree. And you'll note on the right that we have this particular diagram, which is very, very important in terms of our tree-like understanding of evolutionary processes. And we have the keywords here when we're thinking about phylogenetic inferences. We think about the evolutionary relationships of different samples. And therefore, how to read different types of trees that occur is very, very important. So you might be most familiar with trees like this, where think about the branch lengths here, where the horizontal distances are meaningful. They denote the mutations that occur on this lineage leading to this orange sample, whereas the vertical ones are just cosmetic. So we can scale this up and down depending on the number of samples we have and how do we want to represent this diversity that's present. So those vertical lines are cosmetic and horizontal lines are meaningful. And you note that if we use the branches like we saw in the slide before, with this kind of approach, that can work as well, but it may sometimes be more difficult to interpret properly what's going on in terms of measuring them across this x-axis scale. So therefore, representing them as horizontal and vertical bars is probably the most effective approach. So what we have here then are nodes denoting the particular samples shown in green here, and the internal nodes are shown by these blue spots, blue dots here. So these are external branches here for each particular sample, and these are internal branches that connect the two different blue dots. So they're inferred from the tree, their prediction, they're not necessarily the absolute truth, and the external branch again, their links, the prediction that there is an external branch here is obviously going to be correct, but their links might also be incorrect too. You see here this purple node, that's the root of this particular tree, and there's some sort of rationale behind how we construct that, which we'll get to later. So if we look at this tree, what we can say is that vars 1, 2, 3, 4 are what's called monophyletic. They have a single ancestor, and they form a single clade. If you look at vars 8, 9, 10, they're paraphyletic, so they don't share a single ancestor to the exclusion of all other viruses. If you look at vars 6 and 8 alone, for example, they're polyphyletic because they come back to different ancestors, and there's a sample in between them. Vars 7 there, that means that there's more going on across these different lineages than just monophyly. So if you look at an example from SARS-CoV-2, we have here time on this x-axis instead of the numbers of mutations. I know that the y-axis here is just cosmetic. And we can add on different colors representing different clades or groups that are of interest. So for example, if we look down here, there's several different delta clades down here at the bottom that form a single group, and they're different from the different Omicron lineages over here. So we have these horizontal lines representing genetic distance, so we have the vertical lines that are cosmetic. We have the x-axis representing genetic distance or time. We have our different nodes here represented by the different isolates, and the edges connect those isolates together based on the tree that we've constructed with all these samples. So we can represent this tree in different ways. For example, we could make it radial, as shown over here. There's also variants on that called polar trees and fan type trees as well. But radial is probably the one you're going to use most. If you have a lot of different samples, you want to add that metadata onto there. We also have unrooted trees, which are really great if you have a collection of samples for which you don't know the root, although you might hypothesize that it's at this point. And that therefore you just want to represent the diversity that's simply present in these particular samples. So it's a less biased method if you're looking at a bundle of samples. And you can see that here then for the SARS-CoV-2 data, we've got the root specified here on this radial tree where we're showing the time here shown by the different contour lines that are present. Also effective for representing diversity is our networks. So they can represent these unresolved internal branches where we have, say, across ABCD here, differences across the alignment. So there might be a portion of the alignment where A and C are more similar to each other, but then another portion of the alignment where A and B are more similar to each other. And vice versa, C and D might be more similar to each other at the start, but then A and C might be more similar down here, and vice versa for BD. So there might be two different regions of our alignment that produce this kind of internal conflict, and therefore a network can be a good way of representing that, especially for recently collected samples where there's more likely to be this kind of heterogeneity. And our way of getting around this then is to simply reconstruct those different sections separately. So you might have more than one tree representing a segment of DNA. So more terminology then, we can think about this tree here where we have a root and a branching point. A taxa that's close to the root of the tree is sometimes referred to as a basal taxon. We might have instances where we have two samples that are closely related. They're sister taxa. They share a single common ancestor. What else do we have? We have polytomy, and this is where we have a branch with three or more lineages emerging at a particular point. So that can be because they're a similar pattern of mutations or inconsistency in the alignment such that their average number of mutations is similar across the different branches, or they've similar external branch lengths. There can be reasons why we get some sort of polytomy across that. So it's worth noting in trees that when we have a tree, that nodes can be rotated, so we can switch the bat, camel, and human viruses here. We can rotate them vertically and have the humans up here and the bat one down here. It makes no difference in terms of the tree. So the structure of the tree is cosmetic, but it's notionally logical to sort the trees by the genetic distance to have the ones that are most divergent, either the top and bottom, and then gradually go down, as you can see here, across this tree. It makes it more easy for a viewer to interpret. What we can then do on a tree like this is try to identify, okay, what's the common ancestor of all these different samples? So we see whale and bat and human and camel are infected, and therefore we could hypothesize, was the bat virus transmitted to humans and to camels separately? Was it a single event? How can you think about the origins of that emergence? So we can say it's likely that the whale gave it to the bat virus, but then we might want to do more sampling to identify maybe there's more samples at this related to this ancestor that can tell us more about that process. So we can think about this, the most parsimonious scenarios that's possible here in terms of the number of interspecies transmissions that might be possible. So if we look at this diagram here, we might hypothesize that there's a transmission from bat to camel and bat to human, two separate transmissions, and they would occupy these branches, these internal branches on the tree. Equally, we could test the other hypotheses of transmission from bat to human and human to camel. So here we have bat to camel, and then camel to human. So that's two transmissions as well at different points on the tree. And then we have this third option, bat to human, human to camel as well. So see, for each particular scenario, we have two different options, and therefore it's not easy to resolve which one is most likely. Certainly the time interval here for this branch is shorter. This one is longer, and this one is longer, suggesting that there's more time available for two separate transmissions, in my own opinion here. So that would be my hunch, but it's not, the data doesn't necessarily answer that. We'd have to do more sampling, really. So we can look at different trees like this, and what we see here are bootstrapping estimates which test the robustness of this node. So this is on a fraction basis going from 0 to 1, where 1 shows 100% for support for this particular topology of virus 1, 2, 3, 4 in terms of the basic topology that 1 and 2 are more related to each other, and then we have 3 and 4 are more related to each other, and then 5 is the outgroup. It suggests that all the sites that are being aligned here agree with that basic topology. The distance might be different, but that's the basic topology that fits all those particular sites. So what bootstrapping does is estimates the robustness of the alignment by resampling across all the different sites that are in this alignment. So if there's 100 sites, what we might do is randomly resample from that 100 sites with replacement. So sometimes sites might get oversampled, some might get undersampled. So that's why here, where we have a shorter internal branch, we see here that not all sites agree with that 35% don't, and therefore the topology of 1, 2, 3, 4 here is less well supported. So you say that for the virus 1 and 2 here, that is very, very high, and therefore that's probably robust, whereas this value of 0.65 is not sufficiently confident to really tell us that this is necessarily the true topology, and certainly does vary across the phylogeny. And we see another pattern here where we have 0.51 for this particular split. And therefore, we can use this then to test the topology of the tree and also do things like infer the outgroups, which is complex because we want an outgroup that's somewhat similar to the samples, but not too distant as well, where most of those substitutions have occurred. So this inference of an outgroup is also tricky. We can apply these approaches, for example, to this instance here where we have 2008 outbreak of I think it was avian or no type of flu. We've got EMA clades 1, 2, and 3, and we can ask, are they particular clades? So you can see here we've got a cluster altogether, and we see the bootstrapping estimates all show 100 for that particular clade, suggesting that this tree topology is robust. If we look at EMA clade 3, we have a bootstrapping value of 100 again, suggesting that's robust. If we look at EMA clade 2, we have this collection of samples that form a single phylogenetic clade. We see the support here is 83. So what we can infer from that is that the branching of 2 as more distance from 1 and 3, that maybe is most likely, but not necessarily categorical. So you can see that each of these is likely to be a clade, but the extent to which that's certain for clade 2 is lower. We can say that. So we don't know, for example, this divergence sample here, could that fit into a different point in the tree? So we're thinking then, is this 2007 outbreak genetically distinct? Are there 3 different clades? Do they have one single ancestor, or maybe 3 different ancestors that are closely related? Another example here is given from PB2 gene region from flu samples. So you've got the avian H1N1 are in green, shown here, and you can see the nodes are green as well, and then the others are in red, green, blue. So you can see them mixed up here. The current influenza pandemic is shown by the triangle at the top up here. We can ask, in what year did this group, the blue triangle, share a common ancestor with the avian H1N1 samples? So we can track back here to find out based on the tree, when did they have, when was the common ancestor of all the different samples here, which looks like it was 1971, but with uncertainty here represented by the orange bars, which appear to extend maybe up to 1976 and down to 1972, perhaps. And then if we look at the ancestry of the most recent common ancestor of this group with the blue group, we can go to this branch here, where you have an estimate of 1989, but that appears to extend to 1992 and down to 1987. So these are typically credibility intervals representing the highest posterior density of the estimate of this time here. So these bars represent a kind of a distribution that peaks at that point represented on the graph, but may have asymmetry associated with this, and gives us a sense of uncertainty associated with this particular estimate at this time point. In another just final example, then we can think about in what year did the triple assortment swine lineage originate? This is from the PB1 segment of flu again. So we got our triple assortment swine out here. And what we can do is then look back at our ancestor in this particular tree and try to identify, so that nose appears to be part of it. When did that occur? So that appears to be 1995, with maybe 1994 to 1996 as an option. And we can differentiate that from the human H3N2 clade here. And you can say in what year did it have a common ancestor with the human H3N2 lineage, which appears to be around a year before, so 1994, but with credibility estimates extending maybe to 93 and 95 as well. And we see the common ancestors back here for the whole tree in 1987. So these are all examples of how we can assess phylogenetic data from different trees that are drawn and kind of derive information from the data represented here. So please see our next video, which will look at sequence alignment and tree construction optimality criteria.